introduce yourself first.
Good afternoon, everyone. I'm Tapasya, and I'm from Master's College. So today I'm going to introduce our today's speaker, Professor Tapasya. She's an associate professor at Delhi South Campus. She did a bachelor's from Delhi University in zoology honors, then went on doing a master's in biochemistry from Untitled University. She did a PhD in genomic instability in astrocytic tumors and cell lines from AIMS, that is All India Institute of Medical Science. If we talk about her work experience, she started as a young scientist fellow at AIMS, then became the research associate at AIMS. Later, she was the deputy assistant director at National Center for Disease Control. From 2009 to 2017, she worked as an assistant professor at the Delhi South Campus, and now she's an associate professor at the same Delhi South Campus. She's been honored with lots of awards. I would like to name few of them. Like she's been awarded with Junior Research Fellowship by the Lady Tata Memorial Trust, then Senior Research Fellowship by the Government of India, Women's Excellence in Research Science Award by Department of Science and Technology, and there's a huge list and it would be a lot of time. So there are many more awards. And apart from these, she has written a lot of research papers, review papers, and even a lot of. She's been authored one of the books like about biochemistry, and she has written chapters in books and ebooks. The lab she works upon cancer biology and genetics, and the lab works upon research that is pertaining to both basic as well as the translational work in cancer, and they often work with other labs in collaboration. So we are very eager to hear from you, ma'am. And today she is going to talk about epigenetic regulation in plants.
Thank you very much for that introduction. So this lecture is a very basic lecture. So my apologies to the faculty's leader, but it was meant for you people, so it's made that way. Please feel free to stop me anywhere in the middle and ask anything. I'll try to bring a couple of things and try to make a cohesive story out of them. In this process, I might cut out a few things and I might give an overview on a few of them. And but as long as you understand a couple of points, I think my job will be done. So we're talking about a couple of topics here which I feel have not been covered until now in the lectures. One is about genetic regulation, and then also about gliomas, which are a subtype of brain cancer, which is what we work on. So we work on many things, as was mentioned. We work on different cancers, but mostly in cancers where hypoxia, which is a feature of the tumor microenvironment of the cancer, within the cancer, you can have pockets which have less of oxygen supply, less of blood supply. So we work on that, and we don't really look at any one cancer. So most of the solid tumors, this will be relevant, and therefore even in our lab we work on lung cancer, we work on gliomas, we work on GI, and whatever comes our way. If it's to do with hypoxia, usually we take it. So just to give a very brief overview, and I can see one picture is not turning up here, but that's all right. If you look at the basic understanding, which I'm assuming you would have heard of some of these terms. You would have heard of genomic imprinting, which basically means it's a phenomenon in which some genes are switched off maternally and some genes are switched off paternally. And there are very few such genes which undergo this parental specific switching off. Okay? And this is a phenomenon which is associated and is called as an epigenetic phenomenon, largely because not because of how it is happening, which is also there, but also because you are getting this parental specific choice sort of which is made, which is inherited in a way. So individual after individual would tend to get a certain set of genes which will be imprinted on the maternal chromosome, and those which will be imprinted on the paternal chromosome. The process by which it happens, which involves DNA methylation, non-coding RNAs, and a lot of other regulatory mechanisms is what makes it into something which comes under epigenetic regulation. You would have also heard of transgenic inheritance, although this is not very well worked out as an epigenetic phenomena, but there are enough reports which suggest that, if a person or if maybe a pregnant mouse is eating a certain kind of food, then in the developing embryo, right? In the developing embryo, in the progenitor germ cells of the developing embryo. So here I'm going one within. Okay, so you have the mouse, you have the developing embryo, and that embryo will have its own set of gonads which are being formed, and that becomes your progenitor germ cells, the germ cells as they're called. And it has been seen that if you make a difference in the diet of that pregnant mice, then you will have a subsequent generations whose gene expression can be regulated by it or be affected by it. Now, this can be seen in some experiments such as this, where you can see a particular gene called agouti, which, if it undergoes methylation, whether it is hypermethylated or hypomethylated, that means a lot of methylation on the agouti gene, or hypomethylation, very less methylation on the agouti gene, can actually make a difference in the coat color which comes about in the mouse pups. Okay, so people have used such markers. It's like a marker gene because you can see a change in the phenotype, right? I mean, that is what our whole perspective is. You find a change in a gene, you should be able to see that change. And in case of epigenetic phenomena, I'm giving you some examples where you can see the phenotype changing with a change in the nutrient content you're having. Yeah.
You said like methylation status, hypomethylation and hypermethylation.
Yeah.
Is there any basal level of methylation you are talking about?
Yes, there will be a basal level of methylation.
Like
That, for example, an agouti gene or in a particular backdrop, genetic backdrop, your agouti gene may be expressed and could be giving a certain color, or it could be repressed and therefore giving a certain coat color. In the next few slides, you will get this in a little more detail. I'll just come over.
methylation in eukaryotic DNA.
So every individual will be born with a certain set of methylation marks on them. Okay, and I'm not talking about genomic imprinting here, which they definitely are, but also for other things. However, over a period of time, it has been seen, especially with twin studies. That means you take twins and you put twins under different sort of environments, maybe different type of food that they're having, a different place. Maybe they're, one is in Delhi, which is like a highly polluted, and maybe I'm just taking as an example. Okay, but on twin studies, it has been seen that even though twins come with an exactly identical genetic makeup, that means whatever is baseline is going to be the same. Your environment can influence expression of certain genes, and that is where you see that difference actually come up very, very well if you talk in terms of getting your controls right. I'm sure over this period of time, things would have been emphasized on how important control are. So I appreciate that a baseline methylation will have to be defined. So the last one is something you would be most familiar. Samne kara hai, bar body samne banai hai, right? And cheek cells for the women, you can see one X chromosome getting packaged and deposited towards the periphery of your cell, as in shown by with that black arrow. And this process of X chromosome inactivation is also an epigenetic phenomenon. I'm just going ahead with familiarizing with some of the ones which are probably you are already very familiar with, but just to start off. So if I had to pick up X chromosome inactivation in a little more detail, I will pick this up to try and understand a few other concepts along with it. Now, if you look at this, what it tells us is that suppose in my this X chromosome inactivation is a random is it a directed one? Does the same chromosome get inactivated?
Random.
Random. It is completely random. Correct? So what that means is that in every cell of my body, as well as every woman who's sitting over here, every cell would have taken a decision to inactivate one X chromosome. Correct? And this is a totally random decision. Now, imagine if your X chromosome was coding. Suppose I had an X chromosome. In my X chromosome, I had a coding for it's just completely hypothetical, for a marker gene which gave me, say, a certain color, right? Now, what that would mean is that if I start looking at my skin under the microscope, I should be able to see a patchy formation of each of my skin cells, because some would be exhibiting one and some would be exhibiting the other, which is like a mosaic. Okay? So you would get to see it. Now, it is very difficult for us to see, look at it. We don't have, well, we do have a lot of pigment genes and so on. So, if we have to see it in some way, in some anomalies and in some diseases, you can actually see it in women too. You can see stripe pattern actually emerging, but it's very rare. What you see more often is something like this. And this is a tortoise shell cat, right? Now, in a tortoise shell cat, you will be able to see a patchy brown and an orange color all over. You would have heard of this more in terms of calico cats, and maybe heard of it in calico cats. But calico cats and tortoise shell cats are slightly different. Now, all of the pigmented genes that are controlled by this in this cat are done at two levels. There's no one gene which is determined. The base color of any cat is determined by a set of genes which is present in the autosomes. However, they have one type of gene which gives you an orange color when dominant, which is present on their X chromosome. So if that dominant gene is present, right, and it is being expressed, then it will give it an orange color. Now, if that is not expressed, or that particular allele is not dominant, you have a recessive form of that, then it will give whatever the base color it was supposed to give. In this case, the base color is dark brown. Okay, so this tortoise shell cat is actually, when you see something like this, it means that this particular cat is heterozygous in nature. Okay, has either has one dominant allele and one recessive allele of this orange gene which is present. And because it is present on the X chromosome and randomly will be inactivated across different cells, in some places you'll see the orange color, and other places you will see the baseline, whatever the color is. I hope that's clear for everyone. So this is what you see. And if you look at the tortoise shell cat at the bottom, is going to be starting with a heterozygous. You cannot see this pattern in the males on the left top side. The males will never have, right? They will never have. In some cases, it has been seen. But what do you think has happened? They would be having another defect. Can anybody think of what could possibly be the defect in a male? If a male cat is showing a calico, or I'm not even talking about calico here because that involves a third gene, a white one. We are not talking about that. But if you see this tortoise shell, can you see it? If you see it in a male, what do you think has happened? Any guesses? Wonderful. Klinefelters, right? Double X, Y. Right. So that if that happens, then even in the base, you will see this kind of a pattern. That's rare. So if I'm talking about, I'm just coming down to, I thought this was an exciting one to at least to clear up some of the basics. X chromosome inactivation is, we don't do it in our lab, but it's a beautiful example for anybody who wants to learn about epigenetics on how people have traced over a period of time, how X chromosome is shut down. It's quite complicated now, and we are not getting into that. So here what I'm trying to talk about is gene regulation in general, and we would be talking about the expression of genes which are based on sequence changes. You can have changes in the genome based on where your transcription factor is binding, whether there is an absence or presence of a gene, whether there is a mutation present, and so on. And then within that, as a part of gene regulation, is the epigenetic phenomenon, where you can see some of these classic examples such as histone variants, histone modifications, DNA methylation, microRNA, long non-coding RNA, and actually many other non-coding RNAs also come under it. All of these come under the epigenetic control. Why? Because in all of these, the variation is never at the sequence level, right? Nothing changes. Your A, G, C, T, whatever is the pattern is not going to undergo any change. You can have different kind of changes. For example, an entire segment can go and lodge itself at another place where it's a heterochromatic, which does not express genes very easily. That can happen, but still in that segment there has been no change in the sequence itself. So anywhere where you don't have a change in the sequence of the gene, and yet you are able to control the expression of the genes, would come under epigenetic. And it's vast. Some of the examples is what I've given. In plants, you can have hormonalization. In C. elegans, you can have lineage genes which are regulated by miRNAs, right? In case of Drosophila, you can see the red and white eye. You can see that, and in fact, it's a very long. Don't even get me started on that. I won't stop. Okay, so you have that, and then X chromosome inactivation is one I've already given you. A very important one was what Barbara McClintock observed in maize, where the speckled corns, and additionally, along with her work, there were other people who worked on paramutations. Okay, in maize, paramutations in different organisms actually, but also and also mat locus, the mat switchover locus in yeast. Here are some of the examples. But what we are talking about is gliomas. And glioma is nothing but a type of a brain tumor. So I'm going to try and talk about two aspects of glioma disease pathogenesis. There can be many more, but I have cut short so that we can understand at least two aspects well. So this is a very common pediatric tumor, and you can have many types of gliomas based on the cell which is involved. Like in this case, gliomas is a glial cell, but you can have many other type of brain tumors also. You can have oligodendrogliomas and others. In this case, the disease pathogenesis that we talk about will involve gene regulation as well as some epigenetics. If you look at the genetic aspect of it, I wouldn't say it is very well characterized. That means as a genetic disease, it is not as well characterized except for a few known mutations, and these are here. ERCC1 is a gene which is involved in DNA repair and is often implicated and known to be mutated in patients who have glioma. And then you have PP53. P53 you would have all heard of, which is a very well-known tumor. Anybody who's not heard of PP53, everybody has heard of it, right? In the cell set, anybody who's not heard of it, you can just ask me, I'll tell you. Okay? And IDH1 and IDH2 are a set of genes of isocitrate dehydrogenase 1 and isocitrate dehydrogenase 2. Again, these are known to be implicated. This is a part of your PCA prep cycle, right? Then EGFR is something you would have heard of, which is the epidermal growth factor. Know about this, especially to do with other cancers such as lung, breast, and so on. And finally, MGMT, which is a methyl transferase, methyl guanine methyl transferase. Again, this gene is also very well known to be associated in gliomas. Now, all of these genes have somehow helped us in better profiling the cancer, which means they're trying to understand that, okay, I have this particular cancer. Can I have it under a certain profile? And maybe some drugs can be more accessible to me based on my profile as compared to the other. That is the reason why people do it. People want to give therapies which can be more directed so that you don't waste months giving a therapy which will not work. So people want to characterize it. They do the biopsy of the cancer, they try to take out the tissue, they do the molecular profiling, they look at the genetics of it, they try to look at expression of certain genes and say, okay, this person has XYZ, and we have a drug which will work in this patient, and I should be able to give it. That is the reason why people do molecular profiling in so much detail. Otherwise, why bother? Right? There are so many drugs which we take. But advancement in medical sciences is sort of gone hand in hand with the advancement in such methods of molecular profiling and pathology.
Yeah. How will you get these markers in the particular concept?
You take the biopsy, and then, so there are two ways of doing it. You take the biopsy, or if a biopsy is inaccessible. Can you tell me why biopsy may be inaccessible? Can there be a reason? Ethics. For some, yes. But can you think of a more, like, we leave ethics aside, and then apart from that?
The patient.
Then, you can, if you have better tools, yes, you may be able to do a lot of things better. But here it is a concept of access.
And the patient is
Blood cancer? Yeah, what about it? I'll just listen to it. What about it? Leukemias of blood cancer. In that case, we want blood or plasma as essential rather than tissue. So what you're saying is that in case of leukemia, it is easier. That is what you're trying to say, right? Absolutely, it is easier. One can determine it much more easily. You can take it out or whatever. That is fine. Yeah.
But the gliomas are in the case of brain and spine.
Ah,
The patient is living.
Absolutely.
Maybe we can.
It's because you don't want to be operating on the patient twice for once for taking a biopsy and the second time for actually conducting a surgery. So in many cases, you do it once and you do the surgery based on your imaging that you have, and then you can always take the tumor that is taken out, you profile it, and decide the next course of action or treatment accordingly. Remember that for most of the cancers, the first treatment is surgery. If surgery is not possible, then people use chemo, and then they either try to reduce the size of the tumor, make it smaller so that it can be taken out more easily by surgery. So it goes like that, right? So in this case, at least in case of gliomas, you really don't want to be able to. So if you have a germline change, what is meant by a germline change?
In the genetic space.
Where can you see germline changes?
In the
In the
progeny.
blood? Progeny to wo hai. That what you're saying is fine. That is the inherited. But if you take a blood sample from me right now, and what you were calling as baseline, right? So if you see a mutation there, would you consider that a germline mutation?
That may be acquired.
It may be a.
It can be acquired mutation. Which is showing in your blood, right?
Yeah. What
you may acquire it in your lifetime.
Huh? Not a germline.
Not a germline.
Why would you say that?
Right now, you can have a mutation through X-rays in your bone marrow. Your hematopoietic stem cells can.
Okay, but remember that those are all cases which are quite extreme. Your blood is constantly being renewed. You are constantly replacing it. So more often than not, what you're seeing in your blood would end up being your germ. Would end up being your
Because that X-ray and all, you are seeing it only harm a particular tissue or subtype. It will not come to the blood.
your bone marrow will constantly be replenishing. So your blood, your blood cells are actually replenished for how many days? 120 days, right? But if you, so that is the reason why that's the reason why people use biopsy. Why would you need a biopsy otherwise? Sometimes you don't get somatic mutations in your blood. You get somatic mutations in your cancer and in your tissue type. And that is what is usually considered as somatic mutations. Okay? So in 2021, these gliomas were actually classified based heavily on some of their epigenetic marks. Now, you do have a lot of other things which will also come up, but this is what I'm just taking as a baseline, that you have the status of your epigenome keyed in the current classification of gliomas. So obviously it's something important in it. And you have these various classifications. So the newer classification that has come about, I'm not going to go into the details of each one of them. If anybody is interested, I can share papers with you. But I want to draw your attention to a couple of things. Okay, one is, if you look over there, you have certain mutations, and in the arrow marked on the right top, certain mutations, presence and absence of certain regions of the chromosome, mutations of certain genes. If you look at PP53 mutation as being one of the genetics which is helping in, you also have 1p19p, which is again location on your chromosome. 1p location is a part of your chromosome, 19p is again. So there is a co-deletion in both of them. So this is very much the genetic aspect of it. Deletion, changes, all of this are the genetic aspect of it. But if you look at the sixth one, you actually have DNA hypomethylation, which has also been considered. So what has been seen is not only this DNA hypomethylation, but CIMP, which is a mutative phenotype based on your methylation profile, which has also been considered and taken as a part of all of these. And you can see that there are other things, aspects of this, which I'm not getting into right now. Just remember this much, that you have methylator phenotypes, mutator phenotypes, as well as methylation phenotypes, which can help you determine on whether it is a good, I would say good, prognosis. So hard to say good cancer. There is no good cancer, but a better prognosis compared to another one. So you compare different rates and you say, okay, if I give this treatment, then I'm going to have a better outcome and more chances of recovery as compared to a second one. In case of gliomas, you have low grade and high grade as something which is often seen. And what has been found is that you also have those which directly develop as primary tumors and become highly aggressive tumors. That means with a poor prognosis. And then you can also have a slow progressing one, which moves from a lower grade towards a higher grade. And it has been seen that there is quite a bit of difference into the role of methylation in all of these. So the ones which develop straight on and becomes quite aggressive does not seem to have the methylation profile as making much of a difference. But the slow progressing one does. Okay. So we'll talk about this a little more. And in doing so, we will talk about these few genes. I'm going to be talking about DNA methylation as a concept, which I hope you people are aware of, at least the most basic methylation. What is that one? On what residue does that happen? Okay. In DNA, right? We are talking about DNA. Somebody else said lysine. We are not talking about histone proteins here. So please distinguish, even though that is also part of the epigenome. But here we are talking about DNA methylation, not histone methylation, which has a completely different set of proteins, which are going to be either methylating it or demethylating it. So here we are looking at DNA methylation, which is usually carried upon by DNA methyltransferases. And I think a lot of work has also been done by Professor on this. So yeah, you could have actually heard this lecture from multiple times. So if you look at DNA transferases, usually as they're called as DNMP, you have DNMP 1, 2, and 3. They have slightly different functions. Okay, I'm not getting into the details of it, but more or less, they deposit a methyl residue on top of your cytosine. Now, recently, or it's recent if you look at it in my age years. It is recent because around in 2013 or something, people discovered DNA methylation as something which can happen. So here I'm going to tell you a concept which you may or may not be aware of. That you can have a state of methylation in two ways, and a state of demethylation. One is your DNA keeps on replicating, right? And the DNMP is supposed to go ahead and keep depositing those methyl groups, right? And maintain your state of replication. Now imagine DNMP, okay, or there is a mutation that's highly repressed. What do you think will happen to the newly replicating DNA? It will not get methylated and will take up a state of hypomethylation, right? In that case, this kind of a hypomethylation is a passive hypomethylation. Why? Because nobody has done anything. It is just that it has not got deposited. It's a passive hypomethylation. But if I go ahead and give 1011 transferase, which is an active enzyme which is responsible for DNA demethylation, this will go ahead and search for that methyl group and say, I have to pivot this out. So your DNA demethylation as the result of these group of proteins called 1011 transferase of 10, 1, 2, and 3 it basically undergoes an active demethylation. Okay? Both these two concepts are fine. So, in this entire lecture, we are going to be talking about TET, the isocitrate dehydrogenase, which is a very important enzyme in the genetics of gliomas, and something that I am very interested in working on, which is HIT. We will try to see how this hypoxic tumor microenvironment, the genetics of the isocitrate dehydrogenase, whether it's mutant or wild type, along with this active DNA demethylase can regulate the epigenetic profile of a glioma. So let's look at the top one first. Let's look at what is the relation between IDH and TEC1. So if you look at this top, the Krebs cycle, this is telling you very simply where your isocitrate dehydrogenase should actually act. It works on isocitrate, converts it into an alpha ketoglucan. Okay, everybody knows of this. This is what it does. And if you look at the isocitrate dehydrogenase as in wild type, you look at that right, this blue one. This is just showing you that as a dimer, how it comes about and it's how it's positioned. It's an open groove at the top, and it allows your NADP to actually bind it, and it allows the conversion of your NADP to NADPH as well as conversion of isocitrate to alpha ketoglucan. Now, in cases where there is a mutant IDH, this mutation acquires a new enzyme activity. And what it can do is, what it shows is that in its, even as a heterozygous form, so here is one wild type because it exists as a dimer, and R132H is one of the mutations, known mutations. There are a couple of known mutations of IDH. And in this case, it suddenly becomes more, has increased affinity towards NADPH. So what it does is, it acquires a new enzyme activity of conversion of alpha ketoglutarate to 2-D-hydroxyglutarate, and it also can, for facilitating, like all redox reactions do, it facilitates the NADPH conversion to an NADP inhibitor. So your mutant IDH does something new. It is not about only getting rid of the IDH activity. This is what I'm trying to point out. Usually when we talk about this mutant है, तो जो activity कर रहा है वो नहीं करेगा. That's it, right? But here you can see that it does a little more. And so what results in all of those patients and their cancer cells, or even if you do an in vitro analysis of and you put in this IDH mutation, you realize that there is a deposition of your D2 hydroxy. Got it? Okay. And what has been seen is that this IDH1 wild type versus IDH1 mutant, you actually have a better prognosis of the mutant. So people always correlate, you have to correlate whatever you're seeing with the survival, at least in all of these studies. If you don't do that, it will be very difficult to form an association or a correlation with overall survival. And it's very hard to do it. It's not always easy. Accessibility is one, staging is another one. What grade are you studying? What is your control? How long are you actually observing? Is it 3 years? Is it 4 years, 5 years, 6 months? What is your cutoff? But people do it, and people do survival analysis. And what has been seen is that IDH1 mutant has a better prognosis, and both of them exhibit differential methylation patterns, which is because of this discovery that they had, that if the D2HG, such as that seen in IDH1 mutant, is deposited, it actually inhibits TET function. Okay, so TET is I'll just remind you, is the DNA demethylase, and therefore a deposition of D2HG is going to result in a repression of DNA demethylase activities. And what does this TET actually do is also shown here. Here is a 5-methyl cytosine. What it does is, in a stepwise manner, it converts it into 5-hydroxymethyl cytosine, 5-fluoro cytosine, 5-carboxyl cytosine, until it goes back to a state of just cytosine. So this is the demethylation cycle that TET undergoes or takes. So what it does is it goes ahead and inhibits this, and therefore your demethylation is inhibited, and level of methylation that you observe should be high, right? Also, additionally, the DNA demethylase is not the only place where it acts. It also acts on this KDM4, which is a histone modifier. Okay? So it also, not only does it affect the DNA methylation, it also ends up affecting your histone methylation. So KDM4 is a histone methyl transferase, and this also gets inhibited. So together, an IDH1 mutant patient will have a methylation profile which is different from an IDH wild type, and it will end up having probably much more methylation as compared to the other one, as compared to the control. So everybody gets this. So you have alteration. So your IDH1, that means, can regulate your alteration throughout your epigenetic. Because this is random. Remember, this is no one particular CGRN. This is no one particular gene. This is something which can happen on any histone where KDM can act, and on any DNA demethylation where TET can act. Methylation where TET can act. Okay. So this is a very general phenomena which was observed. Yeah.
Like when you showed the previous slide, it was heterozygous.
I showed heterozygous, even in homozygous.
It's a female condition.
It is more severe, but what happens is that severity you can't make out too much. But what people have seen is that it is a better survival, better prognosis.
Better than
I can't, not better than wild type. Better than wild type.
either it is hetero or
Yeah, I don't think there is too much of a difference between whether it has homozygous
The
or not.
point is, once the enzyme will be produced and once the D2HT will be produced,
Yeah,
it will anyway inhibit.
it will.
So the basal level will be enough in the cell to inhibit the TET enzyme.
Yeah.
What you meant is that homozygous will have the same conformation that to the D2HT.
Yes.
Right?
Yes. It performs the same similar. So what did people do? So you have to look at it in two ways. You could of course say that look, prognosis is fine. Let it be. क्या फर्क पड़ रहा है, कुछ नहीं हो रहा है. But that's not how it works. It is never about, as we said, there is no cancer which is a good cancer. You have to take care of the point of doing molecular profiling is that can we come up with drugs which are more specific and better acting in certain molecular profiles. So even an IDH mutant patient would still need your chemotherapy. It would still need the drugs, and they still need to be worked on. So people have been trying different inhibitors for IDH mutant to see what happens. Now this hasn't been a very great exercise because as you can see in these recent years, giving this particular AGI 5198, which is an inhibitor of the IDH mutant, basically it just goes and dodges itself there, where the recognizes the IDH mutant, binds over there, prevents that activity from happening at all. So it relieves the hypomethylation phenotype, yes, because obviously you are blocking the IDH mutant, whether it is actually in a heterozygous state or a homozygous state, but to a much lesser extent as what was anticipated. And what was worse was it confers radioprotective effects. What does this mean? That means if you're giving radiation, that radiation is not working as well, right? If you're giving chemotherapy, it is not working as well because your DNA damage starts getting resolved. So somewhere that methylator phenotype is playing a role in giving it a better prognosis. So through such experiments with inhibitors, they could put two and two together. So what are they doing now? What they're looking at is other agents which may work, which can actually not reduce the survival, but support the IDH1 mutant phenotype in such a way that it further makes it susceptible to chemotherapy, makes it susceptible to radiotherapy. Get it? Or is it confusing? Got it? Yeah. So the last point, what I'm trying to say is that yes, you can come up with an inhibitor, but you would still want to see what is the effect of that inhibitor. In this case, they found that look, it does, it works the way that it's supposed to because this is probably a drug design which is very directed towards an IDH mutant, but it also relieves of the hypomethylation phenotype, but relieving that phenotype did not necessarily help in the survival. And relieving that phenotype still happened to increase the cell resistance to chemotherapy or radiotherapy, which is usually given to cancer patients. It did not help in that. So people are still looking for ways out of this, actually. And a lot of papers have come up in looking at different drugs. So chalo IDH mutant को छोड़ दिया उन्होंने that okay it's leading to a hypomethylated phenotype. Maybe we should do something about the methylation phenotype. So there were some papers which have tried to put in DNA methyl transferase inhibitors. That means you're getting a hypomethylator phenotype because TET is being suppressed. So can we put now a DNMT inhibitor so that the methylation which is accumulating is also suppressed? Will that help? So this is something that people have tried in IDH plasma cells. Quite a success. Azacitidine is another one that affects the activity of DNA methyl transferase that is being used. Decitabine is a more advanced form of this, and this has resulted in regression. That means the tumor tumor which was growing in an implant in a mouse. So what you do is you take some cell lines, put the cancer cell lines under the skin. It's not exactly under the skin, but it's called as the flanks of a nude mouse. The nude mice has a completely compromised immune system. So therefore, it does not recognize it as foreign and allows it to grow. That's how people do xenograft and nude mice studies. Okay. So in this case, what they did was they grew that and then they tried to give azacitidine to the mice. And they saw that doing that actually resulted in suppression of the growing tumor to an extent. So this is something that people are trying now for IDH1 mutated patients. Apart from this, you have, so actually all of these, what I wanted to say here was, all of these drugs, which can change your epigenetic profile, are called as epidrugs. We call them as epidrugs. And these could be of many types. You could have DNMT inhibitors, like we spoke about. You can also have histone deacetylase inhibitors. You can have histone acetylase inhibitors. There's so many of them. There is histone methyltransferase inhibitors, HTMP inhibitors, and so on. So all of these are coming under that one category. And people are, especially in cancers such as gliomas, people are looking at the methylation profile and trying to come up with drugs which can better address these parts. You could call it a precision medicine in a very different way. It's precision medicine not because a particular gene is being targeted, but a particular phenomenon is. So I wouldn't call it any more precision medicine than that. However, there is one example where it does work.
So overall, it's the hypomethylation which is better.
In case of IDH1, mutant, it shows a better prognosis definitely as compared to an IDH. People are not very sure whether this is the only thing. We don't know that.
But both ways, whether you're activating TET or inhibiting PNF1s.
Yeah.
Hypomethylation is there and it's making it better.
It is making it better to an extent.
Prognosis.
But this is one. So it is a bit like, this is the symptom I'm seeing. Maybe tomorrow, if I start looking at it through some other lens, I might find some more aspects to it. So while I can say that yes, it has been observed hypomethylation is coming better in this, I can't say that this is the only contributing factor.
And also long term, it may not be. The sense that after all, methylation is a dynamic phenomenon required for regulated genes.
And again, better and worse is like a few months or not.
Unfortunately, I just see both ways it is.
Yeah. So what has been seen is the primary gliomas, where you have a sudden onset of an eye. Whereas the progressive one, which is working from grade 2 to grade 3, 4, that one shows this one. That is why that time thing is coming. So this is the second aspect. I'm running very late. I'll try and speed up a little. Okay. I might have to cut down a few things. But so here IDH mutation, when we are looking at it, when we started looking at it, I haven't been working on IDH mutation. But it's extremely interesting. And what I found was that if you have an IDH mutation, you actually have an accumulation of your hypoxia-inducible factor. Now, hypoxia-inducible factor is a factor which is coming up in your hypoxia signaling network, which again comes if you have lesser amount of oxygen. Then, if you have lesser amount of oxygen, then there is one. So first I'll tell you one cycle, then I'll tell you what happens in hypoxia. Imagine your hypoxia-inducible factor is a protein which, like any other protein, has been produced. Okay. Now this one, in the presence of this protein called PHD, will acquire hydroxyl groups on it. Once it acquires hydroxyl groups on it, it is recognized by this another protein called VHL, which is it's called as von Hippel-Lindau protein. And this VHL therefore binds only when PHP has acted on it to put this hydroxyl group. Once the VHL binds on this, it takes it for proteasomal degradation. So your HIF protein normally, when your oxygen conditions are sufficient, okay, will go ahead and get degraded. So you should not be able to see it in normoxia conditions. Okay? Whereas in hypoxia condition, when your oxygen is lesser, PHP is inhibited. This does not take place. VHL does not bind to it, and your HIF gets stabilized. And in hypoxic conditions, you see stabilization of HIF. So what has been seen is that your IDH mutation, through how it works on PHP, it basically works through this alpha ketoglutarate 2 or 2-oxoglutarate. So what it does is, since there is a depletion of 2-oxoglutarate, your PHP is reduced and therefore HIF is accumulated. Very similar to what happens in hypoxia. So IDH1 mutation shows something which is very similar to what you will see inside the hypoxic microenvironment of a tumor. Does everybody get that? So what we have seen in our work, and this is not very new work. We are advancing on it, but this I'm showing you as maybe a few years back. Okay? So what we found was, and what we started looking at was, we were looking at hypoxia and trying to see, can hypoxia make basically make a glioma cell more aggressive or not? Aggressive in different ways. We were looking at cancer stem cells, and we were trying to see whether hypoxia can activate a cancer stem cell pathway. I hope you all know that having cancer stem cells in a growing population of cancer cells is worse prognosis. It will lead to a poorer outcome, because what cancer stem cells do is they keep feeding into the other cancer cells through at a very highly proliferative rate. So you are going to end up having a very aggressive cancer if there are sufficient cancer stem cells present. So what we saw was that in hypoxia, in the cancer cell lines that we were studying, we were looking at A172, which is the type of glioma cell line, and we saw that, remember the Yamanaka factors? Lot of Yamanaka factors, so you will recognize OX4, NAM, OX2. These are some of those stem cell factors which help in growth proliferation and making the cell more stem cell-like. So what we found was that you can see an upregulation of all of these factors even in the cancer cells. Remember, stem cells are different, cancer cells are different, and cancer stem cells are different. So cancer stem cells are not the same as stem cells. Why? Anybody? Now,
Cancer stem cells
cancer
and
stem cells and stem
make
cells.
क्या difference?
Will they be the same or different? Different. Why? Where do you see stem cells? Where can you see stem cells? कोई तो एक जगह बताओ. अभी तो बात करें. Where can you see stem cells? Stem cells. Do we have stem cells in our body? We
Yes.
do. Many. Very many.
Yes,
Where?
ma'am.
Bone marrow. Yeah, we just spoke about it,
Yes,
right?
ma'am. Expression of the receptors is the
Expression of
the cancerous stem cell may express more receptors than normal stem cell.
more though is a very general word. But what I would say is this. Your stem cells are not arising from a cancer cell, right? So your stem cells, you're starting off with a pool of stem cells, and that is the germline that you started with, right? As progenitor germ cells or just progenitor cells. But if you talk about cancer stem cells, you have taken cancer cells, you have lost some of the cancer properties, acquired some stem cell properties, and therefore you have de-differentiated and then gone back into a de-differentiated phenotype. But when you're talking about stem cells, you don't say de-differentiated phenotype. You are starting off with a pool of stem cells, and that's what it is. So your cancer stem cells will have properties of both. They will have properties which are cancer specific, as well as it will have properties which are stem specific. Okay, so that's what we saw here. We saw an upregulation. I have to rush through the slides. And if you, I don't know if you can see it, but if you look at, so what we could do was, we could put some of these factors and see that in hypoxia, so the top row is normoxia. And you can see that these are just a few cells, right? These are random cells. We have given them factors and de-differentiated them and allowed them to grow and proliferate. So in normoxia, they do not proliferate as much and to the same extent as they do in hypoxia, where they can go ahead and proliferate and form these large colony of cells. So you can see the change in the phenotype in a way, because this is a pro-proliferative phenotype that you see in case of hypoxia. So hypoxia is inducing something more in these cells. I'll skip all of this. We've already spoken about it. So what we found was that you could also in hypoxia, you could see that the activity of these DNA demethylase steps, TEC1 and TEC2 and TEC3, we checked these, and TEC1 and TEC3 were actually also upregulated. So hypoxia was doing something to the epigenome, and we wanted to see whether what it did to the epigenome is what was resulting in the activation of those pluripotency factors. So at this point of time, nobody had actually shown what happens in gliomas and how do they acquire the cancer stem cell properties. So in this case, what we found was that an upregulation of cancer TEC1 as well as TEC3 resulted in an activation of this demethylation cycle, which helped in this pluripotency. So we found aberrant methylation in glioma cells. We could do, I'm skipping a lot of techniques, but I'll just tell you. This is a Western blot, which is shown here. So you can see very specific bands. क्या होता है Western blot में? Primary antibody डाली. Does everybody understand Western? You run it on a gel?
Yes, ma'am.
SGS page हो गया. Why SGS page? Why not any page? साइज तो ठीक है, उसमें भी आ जाएगा साइज. तो वेस्टर्न के लिए why are we looking at that? That's what
even native में भी particularly चला है.
उसमें भी वही है. But कुछ एक
What
property.
does the SDS do?
Yeah, what does SGS do? Primary means what? कुछ और specific बताओ. I am putting IDH1 mutant. ठीक है, वो जो heterodimer था, how will I see it? So I don't remember IDH का वो molecular weight क्या था, but assume 40 something. ठीक है. How many bands should I see when I run?
One band.
One band?
20 kilo डालकर के आसपास. जिसको dimer था ना.
नहीं, single is 40.
No,
So
like you will see one band.
Single band,
Yeah.
40. But mutant की वजह से कुछ आया होगा, नहीं आया होगा, we will not come to know.
Yes.
We will not come to know if there is no change in the sequence, right? But what is important here is you are breaking down the two dimers. It's not existing as a dimer anymore. Imagine if the IDH1 was actually changing its molecular weight, mutant. So you have one, it's a heterodimer. So will it give one band or two bands?
Two bands.
Two bands if it is being recognized by the same antibody. Why? Because it will resolve monomers. Got it? It will resolve monomers.
Yes.
So if you have an enzyme complex, it will break that entire enzyme complex down. You'll be able to see each subunit. So in this case, it is a single tech, and you have to do different antibodies. You need to use different, and I hope all of you know how primary antibodies are generated. No, I don't know. Okay. So anybody wants to give a try?
Like immunizing the animals with the antigen, which will generate the antibodies against them.
Yeah,
Then we can add
basically
the
you have to think of it in concept of look, you want an antibody. What does that mean? जो भी डालोगे कहीं पे उसका reaction तो होना चाहिए. There has to be an immune reaction. So how do you get that?
immunizing the animals.
Now 1 second, I'll take some video.
Causing the infection. You get an immune response.
But here you are trying to generate antibodies. Infection तो नहीं है, but एक major चीज क्या चाहिए? हाँ? Antigen होने के लिए क्या होना पड़ेगा? Protein, yeah. I get it. Okay.
Adjuvants and we can add.
The basic requirement is कि human से derive करके cell human में ही मत डालो. Yeah. If you want a reaction, you have to take it out of one organism and put it in another one, which is going to recognize that as foreign. Right? Got it? So human का protein उठा के you can put it in a mice. The mice will generate antibody. Got it? This is the primary requirement. This tells you the entire concept of antigen and immunity. So this is in immunohistochemistry or immunofluorescence, if you could say, not histochemistry, fluorescence, and where the secondary antibody that was used was stacked with a fluorescence, which is the green one. This is the DAPI stain, which will stain all your nuclei. So you can co-localize your protein of interest with a dye that is recognizing only the nucleus and say that, okay, my protein is also in the nucleus or my protein is in the cytoplasm. So you use multiple such dyes to help in localization of your protein of interest. Has everybody understood this? I'm guessing you may have heard about it earlier, so I'm not guessing. So on the left side is a densitometric analysis of a dot plot that we did. It was a very simple dot plot. All we saw was if you use an antibody which is going to detect 5-methyl cytosine and which will detect 5-hydroxymethyl cytosine, can we look at the amount of 5-methyl cytosine in normoxia and see what happens in hypoxia? So if you see over here, in normoxia, whatever signal it is showing is your baseline, and in hypoxia, it decreases drastically. So in case of hypoxia, through PET, you have this loss of 5-methyl cytosine and a gain of 5-hydroxymethyl cytosine because that is the downstream product of PET. Okay, got it? I'm not getting into TNMPs now. So I'll just explain from here and then probably just stop after this. Maybe one or couple of more slides. But what we were trying to do. So if you look at this is an OX4 promoter region, right? I hope you all know what is a promoter. क्या होता है वहाँ पे?
ट्रांसक्रिप्शन मॉड्यूल सेकंड ईयर में पढ़ चुके होते हो. ट्रांसक्रिप्शन तो सेकंड ईयर में पढ़ चुके होते हो.
Binding site for the
Okay, I'll just take hers first. Modulator. Okay, I'll let you put under. We're talking about lab and crypto and all, which you would have heard. Okay. So basically, a region which is upstream of your transcription start site, where certain factors can bind and regulate your gene expression. Correct? Why will I not call this epigenetic as such? Because your transcription factors are known to bind to specific regions, right?
Yes.
And there is a sequence specificity there. Okay, that is why usually it's not. That's why I distinguish between gene regulation and epigenetic. Some distinction is required. So in this case, we started looking at OX4. We identified a couple of regions upstream of the transcription start site, which had CG islands present there, which we wanted to see, and also where we felt that you could have binding of your transcription factors. And we have analyzed these sequences in more detail using things like CHIP. If anybody's heard of immunoprecipitation, I don't know if you've heard of it, but maybe in the next slide. Yeah, most of the students wouldn't know. So, yeah. So, immunoprecipitation is when you look at chromatin immunoprecipitation as has been shown here. I'll try to give it in a very simple way. I hope you can catch on to that. Suppose I have a gene X, okay, and I want to know whether a factor Y is binding in the promoter region or not. Okay. I know that it has a sequence specificity, so I know that where it is supposed to bind. So, but I don't know whether in my test condition A versus my control condition, do I see any more in test versus that at control on it? So what I will do is, I will chop up, and here in this case, we are talking about protein and DNA interaction, right? Because these transcription factors are going to bind to the DNA. You chop it up into small pieces, you use an antibody, and you scoop out the proteins which are present. Along with the proteins will come some part of your chopped up DNA. Okay? And then you do a simple PCR reaction to see whether that DNA, that protein that you pulled out actually has the fragment that you are looking for in the DNA. Got it? Clear? No? Sort of. Once more. Okay. You chop up this, you have a stretch of DNA. This is a promoter. This is your region that you want to study. This is your transcription start site. You want to see whether a transcription factor TF1 is binding on. What I will do is I will first chop up this into small parts, and it's an entire, not one cell, many cells together. Okay. I'm trying to compare a control and a test condition to see whether there is more binding of TF1 in one as compared to the other or not. Now, how do I look at that? So first, what I do is I make sure this protein is bound properly to the DNA. Then I chop it up. After that, I use an antibody and I pick up this region. The antibody may pick up other regions, but it will get washed off. It is a specific antibody. It's meant for, it's an anti-TF1. So it will pick this one up. Now I have this in two tubes. I have one over here, and this is the second tube that I have picked out for both of these. I will do a simple PCR with primers specific to this part of the DNA, and I will see whether I'm getting a higher or a lower amplification in one compared to the other. Got it?
Yes.
What this will tell me is there more TF1 binding or not? Right? It will show me that because you started off with the equal amount of DNA. You had everything as equal, except you don't know whether the protein there's more protein on it. So that is how all immunoprecipitation experiments are done. Chromatin immunoprecipitation. And we did that. So what we found out was, to cut a whole long story short, that you had some regions which had more of HTK27 methylation, which came down in hypoxia. This is a repressive mark. You had 5-methyl cytosine DNA IP showing coming up in more regions as compared to the other, which tells us some are more methylated and some are less methylated. So we were able to catch the difference that comes from TEC presence and absence translated into presence and absence of hydroxymethyl cytosine or methyl cytosine. I'm not getting into the details of it now. Okay, so I'm going to really, sorry, it's a long presentation, but I'll just come down to this, that somewhere recently it was shown that HIF also directly induces TEC3 expression, and we missed this both. We couldn't report it before, so we got scooped. So we came up with the rest of it on the gliomas, but this is one part that we missed. So let's see what where the work takes us. We are doing a lot of work creating TET1 knockdown, TET3 knockdowns, and looking at what happens in normoxia versus hypoxia. But what I mean is there is a lot yet to be done in the regulation of methylation phenotype, TET-related work, as well as DNMTs in glioma. So that's what we are trying to do. So I'm going to just cut this all. I had a lot of still left. And this is what we discussed today. Just to summarize, we learned a little bit more about epigenetic regulation and what are gliomas actually. We looked at TET and DNMT as something which will regulate the methylated phenotype of the DNAs. We looked at hypoxia and hypoxia-inducible factor as something which could induce these methylation phenotypes, and saw that HIF induces epigenetic changes in glioma, and that TET can actually initiate this pluripotency through these epigenetic changes in HIF and lead to cancer stem cell formation in hypoxia. So that's where I'm ending today. Okay. Thank you.
Yeah,
Thanks for sitting in scientific seminar today. First of all, I would like to thank today's speaker, Professor Tapasya, for taking your time from a busy schedule and giving us the talk here. Ma'am, you explained us things in a very easy manner, and it was very interesting, and it was a pleasure to hear from you, ma'am. I would also like to thank the director, Professor Daman, for giving us this opportunity and the presence and the program to attend, where we could gain so much of information. Lastly, thanks to the coordinators, Aparna ma'am and Meenakshi ma'am, for coordinating the event so well, and to the audience for being so interactive and
You know what it is. Yes, you can ask it.
yes, yes.
And do you work in glioblastoma cells also?
Yeah, this is glioblastoma.
This is glioblastoma. This one.
You can have different type of cells in different type of progression.
I was studying about glioblastoma cells, and but I was seeing 30% of glioblastoma biopsies, there is